Good morning, everyone. My name is Dayami Lopez. I'm part of the Red Corps as a representative for North Carolina Central University. And it's my pleasure to introduce the next speaker, Dr. Bing Xu. He's a principal investigator and assistant professor in our department, Pharmaceutical Sciences, for the Bright Institute at NC Central, and he has a very active lab working on Alzheimer's disease and also looking into different aspects dealing to Alzheimer's disease and dementia like diabetes. I don't want to take too much time, so allow him to talk about his research on ultrasensitive detection technology enable Alzheimer's disease biomarker discovery. Thank you.
Good morning, everyone. So I'm going to talk about AD biomarker discovery and development from our lab and using newest technology. We think it's an enabling technology using some of the very sensitive detection. So I will be focused on those more technical technology aspects, which is different from the previous couple of speakers. So I don't think I need to introduce more background. I think Dr. Ling Wu and Henry did a really great job of introducing the AD molecular pathogenesis as well as the tau biology in particular. So, but I do want to mention a few points which are relevant to my talk. So I think I want to touch on we will touch on the mild cognitive impairment, short for MCI, which is representing the early stage of AD, because that's the stage we are focusing on for early diagnosis. And MCI is further classified into subclasses, amnestic versus non-amnestic. Amnestic is recovering the memory loss. Non-amnestic is different. They're covering other cognitive loss. And there is a need to differentiate between those two subclasses of MCI. And here are the two problems we need to solve, because the importance of diagnosing AD in early stage MCI, but so far the diagnosing MCI from cognitive normal cohort is quite challenging. There is no established biomarker at this point to really robustly diagnose MCI from cognitive normal cohorts. The other aspect I want to mention is the patient with AD, they show clinical symptom overlapping other tauopathies, such as PIC disease, PSP, CBD. These are all tau aggregation related tauopathies. So there is a need of biomarkers to differentiate AD from other tauopathies, which will be critical for targeted therapy. So we're looking at, for biomarker discovery and development, so several biospecimens we are focusing on. Of course, we're looking at the plasma, CSF. I think the plasma is one of the most accessible biomarker sources, suitable for large-scale screening. They are very inexpensive, and they would be perfect to use in the region with the few resources, including like rural areas. However, the disadvantage is they may have a lot of interference from a high abundance of other plasma proteins, so that makes it really challenging compared to CSF. Now, CSF, to the other hand, it is more relevant to the brain. It's directly in contact with the brain. However, it is a little more invasive because you need the lumbar puncture, and it could be also a little bit costly. Now, there are also new other biospecimens, which I will mention the skin, which is showing some feasibility in showing in some other neurodegenerative diseases, such as prion disease, as well as Parkinson's disease. However, the disadvantage of the skin is a lot of things are still poorly understood. How does the skin tissue relate to the brain disease, for example? So my lab, we focus on the molecular features of pathogenic tau or AD tau. And I think as Henry and Lynn mentioned early on, there are dozens of phosphorylation sites across the entire tau molecule, which is a 441 residue. As you can see in panel A, those are listed are the high frequency site in AD patient. So we focus on those. And the other points I want to mention is the post-translation modification sites, including phosphorylation, acetylation, and ubiquitination. They are dynamic. Okay, so there are some of the sites shown in B, they come in early, and there are also other sites, they come in the later stage. So those information which come recently from a group in Boston, published in Cell, provide the rationale for us to choose which site to study. For us, it's very simple. We study all of them in the panel A, the high frequency phosphorylation site in AD patient. Okay, so we did a systematic screening based on those disease prevalence. So we studied over 24 phospho-tau antibodies across the entire sequence of a tau protein. And we identified multiple sites, epitopes, which is very promising looking at to be diagnosis MCI from cognitive normal patient using the brain tissue. And here is a few. I think Lynn already talked about 422. We also have a published study on 198, as well as some other new ones, 356, 396, 404. I think one of a graduate student, Hibak Gandia, will present a poster on 356. So, and these sites, compared to existing sites, they are very promising. Some of those the diagnostic performance are better than existing sites. For example, p-tau181, which is currently in use in clinically. Also coming up, p-tau217, which is considered to be the best currently. So I think we have to do comparison with the sensitivity and the specificity. I think this here showing this, the data showing 396 in comparison, they are almost equivalent to 217. However, I think the separation is slightly better than 217. And it is better than 181 and 270. These are all the data came from the brain. And we want to translate those brain data. Well, first, before we do that, I think we need to do validation. So use the neuropathological staining. This is a collaboration with Jerry Wang from ADRC neuropathological call. So, and we look at the areas, which is mostly a hippocampal area and the adjacent area we consider to relevant to AD in the early development. And we also applied, Jerry applied the machine learning or AI-based technology to do the immunohistochemical staining, quantification, imaging analysis. You can, this is a staining itself, and this is the AI, the technology recognize, and you see how close they are about this technology. And the data from P396 staining demonstrate a higher tau burden in almost all the area except the CA2, they are all elevated in the MCI versus normal controls. So from those postmodern brain study, we want to translate into more accessible biospecimens such as plasma and CSF. And we're using two technologies to do that. One is a ultrasensitive single molecule array called also short for SAMOA technology, which is pioneered by Quanterix, a company in Boston. And two unique features about this technology is it is about 1,000 times more sensitive than ELISA, as which people use routinely in the academic lab. The other, so it can detect up to femtogram per mil of sensitivity. Okay? And the other is it is very user-friendly. For academic lab, can use to develop home-brew SAMOA test for any of the new biomarker you find in academic lab. So we are beginning to do this. And I'll show you one example of those SAMOA study in a second. Briefly talk about the technology aspects of this SAMOA detection, why it is ultrasensitive. So SAMOA study, it's an immune assay. It is essentially it's a super sensitive ELISA. Okay, so all the other things are similar to ELISA. It's using sandwich, you're using beads coated with a capture antibody, then it captures the analyte. In this case, it's phospho-tau in the blood from the patient, and then you have the detection antibody, and then conjugate to a beta-galactosidase, which is an enzyme, which can generate fluorescence upon adding substrate. Okay. Now, one of the unique things is this complex will be loaded to a small disk, which contains over 200,000 of wells. And each well is just large enough to hold one molecule. So that single molecule resolution really dramatically enhances the sensitivity to give like about 1,000 times more sensitive than ELISA. Showing some data about a small study we have done using p-tau181, which is already known as but we study for MCI subclass differentiation. So we're using the matching MCI and plasma samples for panel A and B. Those are for plasma, C and D for the matching CSF sample from the pre-modern patients. And we're showing that both amnestic MCI and non-amnestic MCI can significantly elevated compared to cognitive normal cohorts. Okay. However, amnestic MCI cohorts has a really robust sensitivity and specificity of differentiation. AUC area under curve is between 0.84 to 0.90. The differentiation between non-amnestic MCI versus cognitive normal is mediocre, about 0.72 to 0.75. And there is no difference statistically, I think, between amnestic MCI versus non-amnestic differentiation. So more robust new biomarkers are needed if we want to differentiate non-amnestic MCI from cognitive normal patients, or to discriminate between amnestic versus non-amnestic cohorts. The other method I want to introduce is another new technology. It's relatively new. It's called real-time quick-induced conversion, short for RT-quick. Okay, so this method is different from SMOA. SMOA, you have extremely sensitive detection. In this one, we almost like a PCR of nucleic acid. However, in this case, it's a PCR of misfolded protein. Okay, so you can imagine that. And this is the flow, and it is we get the in this case, it's misfolded the tau aggregation seeds get from diseased tissue from AD patient, and then it uses recombinant tau substrate to amplify it. So you need a highly purified protein to do that. And once it dies, it will generate. It has this process of a long time, 2 to 3 days of 1 minute on, 1 minute off of vigorous shaking, which will really amplify the misfolded protein, and then it generates signal which can differentiate AD versus non-AD. Okay, and now one of the unique thing is this: the seeds you get, you can highly dilute it, 10 to 5th to 10 to 9th of time of original, which will make it highly sensitive. Okay, which is very different from SMOA test. And I will show you 2 pieces of data for the time being. So we're using this method to differentiate. First detect the brain tau in AD versus control. And we did all 6 human tau isoforms, CLN3R, CL1N3R, 2N3R, and 4R variants. In all those cases, we have highly purified protein substrate. We were able to do the RT-quick, and all 6 individual recombinant tau isoforms used as a substrate. They can amplify misfolded tau seeds from postmortem AD brains, and they are different. They are significantly elevated versus non-AD controls. The other piece of data I want to show you is from the skin tissue, which I mentioned very early on. And skin is a little bit more trickier. We have to engineer different tau construct substrate. And in this case, we construct the 4-RCF tau substrate, which showing schematic showing there, or 3-RCF. Those are the repeating area of the mitochondrial, the microtubule binding area. And in both cases, we're showing these AD or rare topsies. They are significantly elevated compared to non-AD controls. So in summary, we are using those 2 new ultrasensitive technologies, and there is additional more. I don't have time to mention on that. They are potentially can be broadly applied to other AD-related dementias, such as rare topsies or other neurodegenerative diseases, including AD, for example, the tau target or alpha-synuclein in PD or TDP-43 for ALS diseases. I want to acknowledge people who did most of the work, Lynn, and as well as a few graduate students. And I especially want to acknowledge our very dedicated collaborator, Jerry Wang from ADRC, neuropathology core, Andy Liu from the clinical core, and when Dr. Zhou and Wang from Cleveland ADRC, Peter Nielsen from University of Kentucky ADRC, as well as some others. So we are, and also the funding sources from NIH and other association.
I'll ask a question. This ultrastructural, ultrasensitive technique, is there any way to look at cooperative PTMs? For instance, are there any acetylation sites that promote phosphorylation events at those areas?
So far, there is no literature report on that. I think that's a very interesting question because you're talking about different post-translational modification, whether they are cooperative or non-cooperative, whether they have some way influence each other. But I think it might be there some way, in terms of configuration, you can adapt that technology to study that problem. But I haven't seen any literature come out to study that.
Really great talk. Thank you. My question is, I might have missed this, but in your Samoa, could you distinguish between amnestic MCI and then progression of AD into later stage with any of your antibodies?
Yes, we haven't. Yeah, we did all, including the AD dementia. We have those data. They can differentiate those as well. We didn't put emphasis on that because we want to detect early, especially the differentiation between MCI from cognitive normal patient, because I think we got advice from Andy say, okay, those are probably most clinically important. They have a difficulty to differentiate because later stage, they have a lot of a tool to diagnose. So, but I think our technique can do that as well.
I just wonder if they come up and then go down or come up and then continue to rise with progression.
Yeah, so I think that's probably site-specific dependent. So there is a lot of site, some sites showing really high in the early stage, like Henry was talking about. And some sites, they're going up and then coming down. So it really depends on which site to use as the probe. But we have not done systematically on that. I think it's a very interesting question related to disease progression.
Thank you.
The RT-QuIC assay is used a lot in the Parkinson's field these days. Is your technology for tau spreading, is it getting to the same level of sensitivity as the commercially ones people are using now for Parkinson's?
Not quite yet. I know the Parkinson alpha-synuclein RT-QuIC is extremely good in terms of sensitivity and specificity over 0.95. Almost like perfect. In our cases, we are now still getting like in the middle range, like 0.7, 0.8. So we have still, I think AD detection using RT-QuIC is much more complicated versus, alpha-synuclein, which is only like one protein. So we still have a lot of things to do.
So, and this may be something you've done and just didn't present, but as you were presenting your data, we were looking at sort of the AUC for different phospho-tau isoforms, right? And so I wonder, maybe this is just like a data analysis question, but have you thought about or have you done this where you just sort of take all of them and throw them into like neural net, like a machine learning like model, right? Because I'm wondering about, if you're normalizing everything to total tau, what is the ratio of phosphorylated tau at one site to another site tell you, right? Like the ratio of two different phospho-tau isoforms. And that sounds like a lot of dividing numbers and putting things into a logistic model. But it might just be easier to throw it all in and let, random forest tell you.
We have not done that. We don't have expertise to do that. I think we need a collaborator.
Talk to Mike Lutz. Okay.
Very interesting talk. When you looked at tau burden, what is the reason you don't see burden in the CA2 area of the hippocampus? Other than that, so regarding the tau burden in the hippocampus, what is the reason you don't see tau burden in the CA2?
Yeah, we don't know that for particular, I think why not CA2, but it's showing up on the other CA1, CA3, CA4, as well as other adjacent area. We don't know the particular reason for that. I think we probably need to do a little more work about to find out why.
Thank you.